Hello and welcome to Progressive Education Society's Modern College of Pharmacy, Nigri, Pune. In this video, we will discuss about the fruit drug Fennel and according to the syllabus of SPPU for SY Farm B. Myself, Dr. Bhushan Pimple. Fennel fruits are usually called as Saunf or Badishi or Fennel. These are the common names, and this is the plant. The plant is very similar to dill or shepu, and the fruits are very commonly used. Biological source is it consists of dried fruits of Foeniculum vulgare, Miller, belonging to family Apiaceae. Previously, the similar family was well known as Umbelliferae family because of the appearance of the flowers, and they appear like a umbel or umbrella like shape. The major chemical constituents are volatile oil, and in the volatile oil, the chiefly it is the volatile oil comprises of anethone, estragol, fenchone, and limonene. Medicinal uses are carminative to expel gases from the intestinal tract. It is used as a flavoring agent, and it is commonly found in certain gripe waters as well. It is stomachic, that is, it stimulates the GI fluids or gastric enzyme secretion. Antispasmodic, smooth muscles. The spasm of smooth muscles can be relieved by consumption of fennel. It helps in digestion not only by stimulating the GI fluids but also increasing the GI motility. Generally, the fennel it appears in various forms, such as sometimes it is roasted and consumed, sometimes the fresh fruits, or sorry, dried fruits, shade dried fruits are used, which are a light green in color. As far as pharmacy is concerned, the shade dried fruits are preferred, which are non-roasted. Now, this is the fennel fruit, which is the having two different halves. Together, it is called as a cremocarp or cremocarpus fruit. But the same fruit on dehiscence gives two equal halves. These halves are called as, each half is called as a mericarp. One mericarp has one seed. Externally, it is light green in color and has a pleasant and aromatic fragrance, sweeter to taste, and the size ranges from 1 centimeter to zero point 0.5 to 3 centimeter. Shape is lanceolate, slightly oblong, and the major special features are the presence, that means the fruit itself is a cremocarpus and has longitudinal ridges. Now these ridges are made up of vascular bundles. There is a major ridge called as primary ridge, and there are two secondary ridges following adjacent to the primary ridge. These are the longitudinal ridges what we can see in the picture. The cap-like appearance is there on the top, known as stylopod, and most of the fennels they do have this pedicel, which is the branch of the flower and which remains even after the uh, formation of fruit. Now, if you want to proceed for microscopy, the fennel is need to be soaked in water for 1 or 2 hours, and upon soaking in water, it becomes soft, and then it can be easily cut. Now this is the way how you should hold the fennel fruit and the blade in a perpendicular direction. Upon cutting in this way, you'll get the transverse section. Usually the transverse section comprises of a pentagonal, roughly pentagonal to star-shaped sections. Now, this is a single mericarp, and if these two mericarps are joined to each other, they form a cremocarp. There is no need for us to have an PS of entire cremocarp. A single mericarp is sufficient because the another mericarp is just the mirror image of the first one. This is the transverse section of fennel fruit. It is roughly star-shaped or pentagonal in nature. And these are the various layers present which can be prominently seen in the TS. The outermost region is called as epicarp because this is a fruit, and you can see the three layers, that is epicarp, mesocarp and endocarp, very clearly under the microscope. The outermost is the epicarp region. This is made up of a single layer cell and it is cuticularized. The central, the white colored region embedded within the star is mesocarp, and this is the major portion of the TS. Next is endocarp region. The endocarp region is present innermost to the fruit but external to the seed. This is the endocarp. If you notice, the endocarp has a parquetry arrangement for the cells. The mesocarp, the fruit, also shows the presence of Vittae. The yellow colored oval Vittae can be seen here, and these are oil glands, schizogenous oil glands. Apart from that, it also has vascular bundles. These are the five vascular bundles which cause the bulging or protrusion so that it gets the ridges. The fennel fruit externally it shows the presence of ridges, and the ridges are formed because of these vascular bundles. They have both phloem as well as xylem in them. Next, as we see at the center region, there are three layers of the seeds which can be easily seen. The seed externally it is made up of testa. It is not tough enough and it is of single layer, and the endosperm cell, the nutritive region for the embryo, the green colored zone, as well as the embryo itself. This embryo is located at the center. Now these three layers together form the seed of the fruit. Besides the epicarp or the fleshy region as well as the seed, it also has the presence of raphe. Now, raphe is one of the central vascular bundle. The vascular bundle, which is located at the central or center of the cremocarp, is called as raphe. And there is a presence of carpophore as well. Carpophore is the material or the line that holds the two mericarp together to form a cremocarp. That means carpophore joins the two mericarps to form a complete or a dual fruit. These are the different layers. If you want, you can take the screenshot so as to maintain. It will help you during the last minute revision for the examination. So these are the different layers and their locations. Now we'll have a look at the staining of this. As far as staining is concerned, usually only two dyes are sufficient. that is Sudan red 3, which stains the oily region. Oily region is present in the Vittae, in the Vittae there is presence of volatile oil, plus there the epicarp is also cuticularized, so it picks up the dye. So if you stain it with Sudan red 3, the epicarp and the Vittae appears to be pink or reddish in color. Now I have purposely shown here bright red color because different screens may have different resolutions of the color, but to understand this is the way how it gets colored. The color is not this intense, but it can be a tint. Next is the fluoroglycinol HCl. Fluoroglycinol HCl helps to stain the lignified tissue. And in the entire TS, lignified cells are present only in the vascular bundles, the 5 prominent vascular bundle, plus this raphe. This is also having lignified tissue. So upon staining with fluoroglycinol HCl and washing with water, the TS will appear somewhat like this. So this is the staining with fluoroglycinol HCl. There is a third stain called as picric acid, which can also stain the oil globule as well as the endosperm, because the endosperm has some amount of alluron grains, but those are not that clearly visible. Now, this is the actual TS of the fruit. Actually, this fruit appears, the TS, actual TS appears in this way. The various different layers are there. We will have a look at each and every layer of the TS. Suppose this is the epicarp, outermost layer. Now you will notice here that the outermost single layer is the epicarp, and upon staining with Sudan red, it gives this kind of color. This is light red, or you can say pink in color. The epicarp, since it is green tissue, the epicarp at certain intervals, it will show the presence of some grooves or depression. Now, this groove or depression is called as stomata in certain books because this is a green tissue and every green tissue performs photosynthesis. And for that reason, the stomata is present in this region. So you can see the stomata also at 40x magnification. That means 400x magnification by using 40x objective and 10x eyepiece. About the mesocarp, mesocarp is inner to the epicarp and has more amount of starch grains into it. So these are the mesocarp cells, and the mesocarp also has Vittae and vascular bundle embedded into it. You can see the starch grains present inside the mesocarpus fruits, mesocarpus layer. This is the zoomed image of the starch grain on staining with iodine. They take up the bluish coloration. So these are confirmed to be the starch grains. As you go inner towards the seed, you will find one more layer, that is endocarp. This endocarp layer is in close contact with the testa or the seed. So the cells which are to the inner side of the mesocarp are called as endocarp. These endocarpus cells, they have a special type of arrangement called as parquetry arrangement. If you see here, every cell is parallel to its adjoining cell, although it may or may not be parallel to other or upper or lower cell, but as far as the adjoining or just the neighboring cell is concerned, it is parallel. We will have a look at the parquetry arrangement. Now this is the design of various designs of parquetry arrangement. You will find the cells are parallel to their adjoining cells, they are not parallel to all the cells but just their adjoining cells, and they are running in a different direction. Now if you notice here, if you look at this particular cells, these three cells are parallel to each other, but the entire group is exactly perpendicular to these cells. Such kind of arrangements are called as parquet tree arrangement. We will see here, yes, this is the zoomed image of the parquet tree arrangement of the cells. This is how the endocarp appears. So this is very typical or characteristic feature of Fennel, that the endocarp is having a parquet tree arrangement. About the vascular bundles, as we have seen, vascular bundles are the major cause for generation of the ridges over the surface. This is the transverse section of the entire vascular bundle. It houses a compactly arranged, this is the phloem, and these two are the xylem regions. You can notice here the phloem has very dense intercellular space, and the xylem is very compactly arranged and not allowing the light to pass through it. On staining, you will find the xylem taking up dark red color, whereas phloem is also stained slightly reddish in color. This is the stain fluoroglycinol and HCl. Vittae, singular is Vitta and plural is Vittae. Now these are schizogenous oil glands. That means the oil glands are made up of cells which have just separated from each other, and they have maintained a well-like structure, and they are still secreting the oil, volatile oil. This gives the fragrance to the fennel. The Vittae can be stained with Sudan red 3, and the periphery takes up the reddish tint. Now these are the three different photographs of images of Vittae. You can even see the presence of oil globules inside the Vittae upon magnification. About the raphe, raphe is an additional vascular bundle which is located at the center. Now this is the raphe, and these are the two different mericarps joined to each other. About the carpophore, this carpophore is a joint or connection that joins the two mericarp. This is located exactly at the center. This carpophore sometimes gets stained with fluoroglycinol HCl. Now this is the on staining with fluoroglycinol HCl. It shows certain lignified cells in it. Now what exactly carpophore is? Suppose this is a cremocarpus fruit which is unripe, and upon ripening or upon dehiscence, it gives out two different mericarps. Now the centermost twig that has two branches, and this is the connective region connection between the two mericarps. So this is called as the carpophore, and in the transverse section it appears in this way. Now as we go inside or towards the center, you will locate the presence of seed, and seed has all the three prominent zones in it. The outermost is testa, which is made up of a single layer flattened cell. This is the testa, and you can notice here the long line of testa, which is one layer thick. And these are the endosperm cells. The endosperm cells are made up of more amount of starch and oil globules. So they can be stained with Sudan red 3. The centermost zone of the seed is occupied by a D-shaped or a semicircular embryo. Embryo is the young one of the plant, or upon consumption of this endosperm food or nourishment, it will develop into a new, it will give out the radicle and plumule, and it will develop into a new plant. So this is all about the fennel fruits.